Alright boys, what we have discussed so far is about types of heterochromatin, and in that we have talked about constitutive chromosome and facultative chromosomes. And facultative chromosomes we have talked about the bar body, that is the two X that is present, out of which one of the X remains inactive. Only one of the X is active in female, XX. So that is what we have discussed so far about the bar body, about heterochromatin, euchromatin, and now we move on to the next part, that is polytin chromosome or Lamb-Brush chromosomes. Polytin chromosomes or Lamb-Brush chromosomes are both much larger in size than the normal chromosome, and this is found at certain stages of the cell division. And since they are large in size, they are more larger than the normal chromosome. They are also known as giant chromosomes. They are of two types: polytin chromosome and Lamb-Brush chromosome. The polytin chromosome was first observed by a man named Balbini. That is why later on we'll talk about the Balbini rings as well. And this polytin chromosome was first observed by Balbini in the salivary gland of Drosophila, melanogaster, and Chironomus. This salivary gland chromosome, because it is found in the salivary glands of the Drosophila, they are also known as salivary gland chromosomes. They are also commonly known as salivary gland chromosomes, and they are larger in size. What happens here is the chromonema divides along the length. The chromonema divides along the length, and since though they divide along the length, they do not separate, they do not completely separate. That is why what happens, it gives rise to number of arms of the chromosome. There are as many as 5 arms in the case of salivary gland chromosomes of Drosophila. It has round about 5 arms. The chromonema, they do not completely separate. So at a point they remain attached, because of which there are 5 arms that is seen in the salivary gland chromosomes of Drosophila. The most important feature that we see here is the dark band and the interband that is present. If you see a polytin chromosome, it looks something like this. It has number of arms like this. The chromonema do not separate, 5 arms. And these 5 arms has a particular type of banding in it. So there are dark bands and interbands. Dark bands and interbands that is seen here in the case of Lamb-Brush chromosomes. Because of which there appears to be a puff here. This puff is known as the Balbini rings. This puff is commonly known as the Balbini rings. So the most important feature here that we need to remember is the polytin chromosomes are much larger in size. They are found in the salivary gland of Drosophila and Chironomus, and it was first observed by Balbini, and it is formed as a result when the chromonema, they divide along the length and they fail to separate, which gives rise to number of arms. There are as many as 5 arms in the case of the salivary gland chromosomes of Drosophila, and there are presence of bands, dark bands and interbands. And it has a puff like this, which is called puff, which is also called the Balbini rings. So that is about the polytin chromosome. Then let's carry on with the Lamb-Brush chromosome. The Lamb-Brush, as the name suggests, it looks almost like the brush that is used to clean the lamps, which looks almost like this. The Lamb-Brush almost looks like this. Lamb-Brush chromosome almost looks like this. Has a handle here. So the Lamb-Brush almost looks like this, and this chromosome also looks like a lamp brush, because what happens here is this, the chromosome gives out the lateral loops like this. The lateral loops like this. They form lateral loops like this, which gives the appearance of a lamp brush. That is why the name lamp brush chromosome. And this is seen in the oocyte or the eggs of the amphibians. This was seen in the oocyte or the eggs of the amphibians. The loops of the DNA. These are the loops of the DNA which spread out and form a loop like that of a lamp brush. That is why the name lamp brush chromosome. This lamp brush chromosome is almost 3 times larger than the polytene chromosome. So you can imagine polytene chromosome is already large. It has 5 extra arms. Whereas the lamp brush chromosome is even larger than the polytene chromosome. The 2 chromatids gives out lateral loops. The 2 chromatids, the chromatids that is here, gives out lateral loops like this. And this gives the appearance of the lamp brush. That's why the name lamp brush chromosome. After the lamp brush chromosome, we talk about viral chromosomes. Viral chromosomes can be both made up of DNA or RNA. It can be made up of either DNA or RNA. Nucleic acid molecules may be single or double helical, or they could be intertwined amongst themselves. Single or double helical, may be linear, that means in a line or in circular position. So that is about the viral chromosomes. Let's carry on with genes. Now the question is, what is a gene? Genes, as Mendel called it as factors. Genes are factors which determine a particular phenotypic trait of an organism. We have talked about Mendel's law of inheritance. In that we have talked about the factors. These factors is what we know now as the genes. After the discovery of chromosomes and chromatids and DNA and all those, now we call them as genes. Now, what is a gene? A gene is an organic molecule which forms the smallest segment of DNA or RNA that carries information for biological function, that is, information for manufacture of proteins or expression of the phenotype. So that is basically the gene. Let's write it down. I think this is very important point, because when it comes in exams, you can write it as what is a gene. Gene is an organic molecule. Gene is an organic molecule. Gene is an organic molecule which forms the smallest segment of DNA or RNA that carries information for biological function, that is information for manufacturing of proteins or expression of phenotype. This is basically what is a gene. So gene carries all the information that is required either for the manufacture of proteins or for the expression of the phenotype. So that is what is gene. Now gene is, we can say, one gene, one biological function. So one gene is responsible for manufacture of one polypeptide, and this one polypeptide may be responsible for one biological function. Biological function means transmission of characters, or we can say carrying out various metabolic reactions. So one gene is responsible for one polypeptide. This polypeptide could be an enzyme, it could be a protein, or it could be structural proteins, or this is responsible for transmission of characters. And one biological function, or we can say biological information, which leads to expression of one phenotype. This is, you can say, the code on which the gene functions. One gene gives rise to one polypeptide. That one polypeptide controls one biological function or information, and that information controls one phenotype. Now, as far as the gene is concerned, there are a number of concepts. We have an old concept which says gene is beads linearly arranged on the chromosome, and it also says crossing over can take place between two genes but cannot take place within the gene. Genes are beads which are arranged linearly on the chromosome, in which crossing over can take place between two genes, but there is no crossing over that could take place within the gene. This is an old concept. Now, after a lot of discoveries, we have come up with a new concept that is called the modern concept, which again says gene is linearly arranged sequence of DNA. In the old concept there was beads. Now we have DNA or genetic RNA. Genetic RNA means RNA, as far as an eukaryote is concerned, it only carries out it is only transcribed from the DNA. But in certain viruses like tobacco mosaic virus, this RNA is a genetic material as well. So that is why we say DNA or genetic RNA. And crossing over can take place not only between the genes, but it can take place within the gene as well. And when we talk about crossing over taking place within the gene, it could be change within the gene, which brings about a change within the gene. So that is a new concept which suggests that genes are linearly arranged sequence of DNA or genetic RNA. Now we go on to the chemical composition of the genes. As far as the chemical composition is concerned, the genes are made up of three main parts, which is, or we can say, genes are made up of nucleic acid, which is an organic acid, nucleic acid, and this remains within the nucleus. The organic acid which remains within the nucleus is called the nucleic acid. It is made up of, this nucleic acid is made up of nucleotides. And this nucleotide is made up of pentose sugar, nitrogen base, and phosphoric acid. It is made up of pentose sugar, nitrogen base, and phosphoric acid. So these are the three units that make up the nucleic acid, the nucleotides, and the nucleotides, number of nucleotides joined together to form the nucleic acid, either DNA or RNA. Now let's talk about nitrogen base first. The nitrogen base. Nitrogen base is again of two types. Nitrogen base again is of two types, purine and pyrimidine. Purine and pyrimidine. Purine and pyrimidine. Purine are again of two types. Nitrogen base. Purine are again of two types, adenine and guanine. And pyrimidine are cytosine and thiamine. In the case of DNA, it is cytosine and thiamine. In the case of RNA, it is cytosine and uracil. That means we can say, in the case of RNA, the thiamine is replaced by uracil. So that is about the nitrogen base. The purine is of a complex type. It has two rings like this. It has two rings attached to each other like this. Two rings attached to each other like this, and nitrogen here and here. So it has two cyclic rings, whereas pyrimidine only has one ring like this. So that is the basic difference. That means purines are larger nitrogen base, pyrimidine are much smaller nitrogen bases. Okay? Purine are adenine and guanine, and pyrimidine are cytosine and thiamine in the case of DNA, and cytosine and uracil in the case of RNA. So that is about nitrogen base. Now we talk about the pentose sugar, the other unit, other segment of the nucleotide, that is the pentose sugar. Pentose sugar, in the case of pentose, means there are 5 carbons, 5 carbon atoms. Pentose, 5. So they have 5 carbon atoms. In the case of RNA, it is ribose sugar. Ribose sugar. In the case of DNA, it is deoxyribose. That means in the case of DNA, there is one less oxygen at carbon 2 atom. That means when we are talking about DNA, the pentose sugar that we have here is like this. 1, 2, 3, 4 and 5. 5 carbon that we talked about. In number 2 DNA, number 2 carbon atom has one less oxygen. That is about the pentose sugar. Now let's carry on with the next part of the DNA, that is nucleotide, that is phosphoric acid, H3PO4. Phosphoric acid is H3PO4, which is same in both RNA and DNA. In both RNA and DNA it is same. Phosphoric acid H3PO4 is same in both RNA and DNA. This phosphoric acid and the pentose sugar forms the railing of the DNA. It looks like this. This is the pentose sugar. To carbon 1, the nitrogen base remains attached. To carbon 5, here we have carbon 5. The phosphoric acid remains attached. H3PO4 remains attached. And to carbon 3, this is 3 because this is 2, 1, 2, 3, 4. So to carbon 3 also H3PO4. Another, you can say sugar remains attached. So the bond that is present here is phosphodiester bond that is present. In between the phosphoric acid and the sugar, the bond that is present here is phosphodiester acid. The bond. Phosphodiester bond. Now, we talk about the function of the gene. Is that clear? The nucleotide is made up of pentose sugar, nitrogen base and phosphoric acid. The phosphoric acid is same in both the cases of DNA and RNA. It is H3PO4 and it is joined at carbon 3 and carbon 5 of the pentose sugar. They form the railing of the DNA double helix. Okay, and the bond that is formed here is the phosphodiester bond. The nitrogen base is again of two types, purine and pyrimidine. Purine is of two types, adenine, purine and pyrimidine. Purine is of two types, adenine and guanine, and pyrimidine is cytosine and thymine in the case of DNA, and cytosine and uracil in the case of RNA. And we have the pentose sugar, which is ribose sugar in the case of RNA, and in the case of DNA it is deoxyribose. These are the basic things which we need to remember. Now let's carry on with the function of the gene. What are the functions of the gene? The first important function of the gene is it gives, with this I will not write. The main function of the gene is phenotypic expression. The gene, the first important function, it gives a phenotypic expression of that particular character. It gives a phenotype. That means the outward expression is controlled by the gene. Then genotype. The function of the other function of the gene is the genotype. The transfer of genetic information from one generation to another by a process called replication. एउटा जेनेरेसनदेखि अर्को जेनेरेसनमा जुन चाहिँ क्यारेक्टर्स ट्रान्समिट हुन्छ त्यसमा चाहिँ त्यो क्यारेक्टर ट्रान्समिट हुँदैन, बट इट इज द जीन द्याट इज बिङ ट्रान्सफर्ड विच कन्ट्रोल्स द्याट पर्टिकुलर क्यारेक्टर. अनि त्यसको लागि चाहिँ एउटा पर्टिकुलर प्रोसेस छ, त्यसलाई चाहिँ भन्छ रेप्लिकेसन भन्छ. रेप्लिकेसन भनेपछि चाहिँ त्यो प्रोसेसलाई चाहिँ भन्छ रेप्लिकेसन. अर्को इम्पोर्टेन्ट फङ्सन अफ द जीन इज इभोल्युसन. इभोल्युसनरी फङ्सन पनि छ अब जीनको. कसरी भन्दाखेरि चाहिँ evolution is a slow and steady process, a change that an organism undergoes from one generation to another. अब त्यो चेन्ज ल्याउनुको लागि केसमा चेन्ज आउनु पर्यो भन्दाखेरि चाहिँ यसमा चाहिँ डिएनएमा, क्रोमोजोम्समा, जिन्समा चेन्जेस आउनु पर्यो. हो त्यो चेन्ज जब जिन्स अनि क्रोमोजोम्समा आउँछ त्यसले गर्दाखेरि चाहिँ एउटा चेन्जेस आउँदै जान्छ, जुन चाहिँले गर्दाखेरि चाहिँ एउटा अर्गानिजममा त्यो इभोल्युसनरी चेन्ज आउँदै जान्छ, त्यो अझै इभल्भ हुँदै जान्छ, अझै राम्रो या राम्रो 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 हुँदै जान्छ. त्यो जुन चाहिँ चेन्ज आउँछ त्यो चेन्ज चाहिँ थ्रु भेरिएसन पनि हुनसक्छ बाई क्रसिङ ओभर या बाई म्युटेसन पनि हुन्छ. तर त्यो जुन चाहिँ चेन्ज आउँदैछ त्यो चाहिँ भित्रबाट, द्याट इज द जिन्समा नै त्यो चेन्ज आउनु पर्यो. सो द्याट इज अनदर इम्पोर्टेन्ट फङ्सन अफ द जिन्स. नाउ द्याट इज अबाउट the three functions, the phenotypic, genotypic and evolutionary function. Now we carry on with the properties of the gene. Autocatalysis. Autocatalysis भनेको चाहिँ एउटा जिनले आफू जस्तै अर्को पनि बनाउन सक्छ भनेको. अनि यो प्रोसेसलाई चाहिँ भन्छ replication. The process by which one gene gives rise to exactly the same copy of it is what is known as autocatalysis. And this is possible because of the gene. Heterocatalysis. अब हामीलाई थाहा छ DNA मा जुन चाहिँ इन्फर्मेसन छ त्यो इन्फर्मेसनले हाम्रो फंक्शन्सहरुलाई कन्ट्रोल गर्नुको लागि चाहिँ फर्स्ट चाहिँ त्यो चाहिँ चेन्ज भएर के हुनु पर्यो RNA हुनु पर्यो. यो जुन चाहिँ छ एउटा देखि अर्को भिन्नै किसिमको त्यहाँनिर मोलिक्युल बनिँदैछ त्यसलाई चाहिँ भन्छ हेटेरोक्याटालाइसिस. सो DNA देखि RNA जुन चाहिँ ट्रान्सक्राइब हुन्छ त्यो ट्रान्सक्राइब हुने प्रोसेस त्यसलाई चाहिँ भन्छ हेटेरोक्याटालाइसिस पनि जिनमा छ पोसिबल छ. अटोक्याटालाइसिस त छँदैछ त्यो त रेप्लिकेसनबाट हुँदैछ. हेटेरोक्याटालाइसिस चाहिँ ट्रान्सक्रिप्सनबाट हुँदैछ. अब स्टेबिलिटी. Your genes are very stable. The information that is transferred from one generation to another has to remain stable. त्यसै कारण त कति मिलियन वर्षदेखि ह्युमन बिङ्स जस्तै छ. तर त्यसमा ड्रास्टिक चेन्ज आउने भने त त्यो पहिला जुन चाहिँ ह्युमन बिङ्स थियो अहिलेको भिन्दै हुनुपर्ने अनि कम्प्लिटली भिन्दै हुनुपर्ने त्यस्तो चाहिँ हुँदैन. बिकज त्यो सर्टन पोर्सन अफ द the DNA, the gene also remains stable. The stability पनि त्यहाँ चाहियो. अब यो स्टेबिलिटी भन्दैछौँ हामीले सँगसँगै चेन्जेबिलिटी पनि छ. चेन्जेस पनि आउनु पर्यो. किन चेन्जेस आउनु पर्यो भन्दाखेरि हाम्रो एन्भाइरोमेन्ट त कन्टिन्युअसली चेन्ज भइरहेको छ. त्यो एन्भाइरोमेन्टसँग कोप गर्नुको लागि हाम्रोमा पनि त्यस्तै अर्गानिजम्समा पनि प्लान्ट्समा पनि त्यस्तै चेन्जेस आउनु पर्यो. त्यो चेन्जेस चाहिँ कसरी हुन्छ भन्दाखेरि इदर बाई म्युटेसन or by recombination which brings about variation. यो recombination जब क्रसिङ ओभर हुँदाखेरि जुन चाहिँ मिक्सिङ अप अफ जिन्स हुन्छ मेटर्नल र पेटर्नल जिन्स त्यसले गर्दाखेरि चाहिँ सानो चेन्जेस आउँछ. त्यो चेन्जेस चाहिँ बढ्दै थुप्रिँदै गएर पछिबाट गएर चाहिँ मेजर चेन्जेस आउन सक्छ. तर त्यो सानो चेन्जेसले गर्दा चाहिँ के गर्छ भन्दाखेरि एउटा अर्गानिजमलाई कुनै पनि चेन्ज एन्भाइरोमेन्टल सिचुएसनमा बेटर एडाप्टेसन पाउनु सक्छ उनीहरूले. नाउ द टाइप्स अफ जिन्स. यो टाइप्स अफ जिन्समा त हामी डिटेलमा नजाउँ. अटोजोमल जिन्स, यो अटोजोम्समा हुने जिन्सलाई अटोजोमल जिन्स भन्छ. यसले एल्बिनिजम भन्ने हामीले जुन चाहिँ पढ्दैछौँ टङ रोलिङहरू जुन चाहिँ छ त्यो चाहिँ अटोजोमल जिन्स अटोजोममा भएको जिन्समा आउँछ. सेक्स क्रोमोजोमल जिन्स अभियसली यो त एक्स क्रोमोजोममा हुन्छ. एक्स क्रोमोजोममा जति पनि जिन्सहरू छ त्यसलाई सेक्स क्रोमोजोमल जिन्स भन्छ. जस्तै हेमोफिलिया, कलर ब्लाइन्डनेसहरूलाई कन्ट्रोल गर्छ. होलोएन्ड्रिक जिन जति पनि जिन्सहरू हाम्रो वाइ क्रोमोजोम्समा हुन्छ जुन चाहिँले गर्दाखेरि चाहिँ हामीले पढेको छौँ नि हेयर पिन्नामा हेयर जुन चाहिँ ग्रो गर्छ ट्राइकोसिस हो त्यो सबै चाहिँ होलोएन्ड्रिक जिन भन्छ त्यसलाई. जम्पिङ जिन्स अर ट्रान्सपोजन्स पनि भन्छ. त्यहाँदेखि लिथल जिन यस्तो खालको जिन चाहिँ हाम्रोमा इनहेरिट भयो भने चाहिँ त्यो अर्गानिजम्स चाहिँ मर्छ. Which leads to this expression of such genes, lethal genes, expression of certain genes which causes the death of an organism is called the lethal gene. Then again also oncogene, cancer causing genes, cancer बनाउँछ. Then split genes भन्छ. जस्तै eukaryotes मा हामीले हेर्यौं भने introns र exons छ. यो introns चाहिँ intermediate genes छ जुन चाहिँले चाहिँ एक्सप्रेसन हुँदैन. exons चाहिँ एक्सप्रेस हुन्छ. यो बीचमा आउँछ जुन चाहिँले गर्दाखेरि चाहिँ त्यो एक्सन्सहरूलाई स्प्लिट गरेर राख्छ. स्पेशली इन द eukaryotes. प्रोक्यारियोट्समा यो देखिँदैन. त्यसलाई चाहिँ भन्छ स्प्लिट जिन्स भन्छ. त्यहाँदेखि ट्रान्सजिन चाहिँ के हुन्छ भन्दाखेरि एउटा प्लान्टदेखि अर्को प्लान्टमा ट्रान्सफर भएको या एउटा एनिमलदेखि अर्को एनिमलमा ट्रान्सफर भएको जुन चाहिँ जिन्सहरू छ तिनीहरूलाई चाहिँ भन्छ ट्रान्सजिन्स. सेल्फिस जिन. यो जिन चाहिँ के हुन्छ भने यो जिन चाहिँ प्रेजेन्ट चाहिँ छ तर कहिले पनि एक्टिभ हुँदैन. यो चाहिँ इनएक्टिभ स्टेटमा नै बसेपछि. रेगुलेटरी जिन. यो जिनले चाहिँ के गर्छ भन्दाखेरि अर्को जिनलाई चाहिँ रेगुलेट गर्छ, कन्ट्रोल गर्छ. त्यसको फङ्सन्सलाई चाहिँ त्यो एक्टिभिटीलाई चाहिँ कन्ट्रोल गर्छ अर्को जिनको लागि. त्यसलाई चाहिँ भन्छ रेगुलेटरी जिन. त्यहाँदेखि आउँछ कन्स्टिट्युटिभ जिन अर हाउसकिपिङ जिन. यो जिनहरू चाहिँ जहिले पनि क्रोमोजोममा प्रेजेन्ट हुन्छ अनि यिनीहरूको चाहिँ थ्रु आउट द लाइफ अफ एन अर्गानिजम यिनीहरूले चाहिँ एक्सप्रेस गरि नै बस्नुपर्छ. बिकज दे आर इम्पोर्टेन्ट फर द सर्भाइभल अफ द अर्गानिजम. बिकज दे आर इम्पोर्टेन्ट टु क्यारी